The first step in mitosis is interphase. This is when DNA is replicated. Next is prophase. In prophase, the centrioles are moved to the opposite poles of the cell and the membrane begins to dissolve and chromosomes form. Then metaphase begins. The chromosomes line up on the equator of the cell and the spindle fibers attach to the centromere of each chromosome. Then anaphase starts. By pulling the chromosomes apart, the chromatids are pulled to each pole. Also, the cell membrane begins to pinch. This is called a furrow. The next phase is telophase. This is when the nucleus forms around the separated chromosomes. When mitosis is complete, the cell completely separates into two cells that are exactly the same. The first step in meiosis is interphase. This is where DNA is replicated. Next is prophase 1. This is when centrioles move to opposite poles and homologous chromosome pairs form. This is also when crossing over occurs. After the nucleus completely dissolves, metaphase 1 begins. The homologous pairs line up in the middle of the cell and spindle fibers attach to their centromeres. Then anaphase 1 starts. The homologous chromosome pairs separate and move to opposite poles. The cell membrane starts to form a furrow. Telophase 1 then starts. A new nuclear membrane forms around the separated chromatids. At the end of meiosis 1, there are two different cells. Then prophase 2 begins. Centrioles go to the opposite poles. In metaphase 2, the chromatids line up in the center of the cells and spindle fibers attach to their centromeres. Next, in anaphase 2, the separated chromatids are pulled to opposite poles and a furrow appears. Then in telophase 2, a new nucleus forms and the two different cells separate into four completely different cells.